Hi, welcome to Neuroverse
a podcast hosted by Clara
and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on methodologies of neuroscientific investigations from a philosophical perspective. And to enlighten us on this topic, we have a special guest, Subham Ganguly, who completed his BSc in neuroscience at King's College London and just finished his master's in brain science at UCL's Sainsbury Wellcome Centre, the SWC.
Hi guys, thanks for having me.
Of course, thanks for joining us.
Thank you so much for joining us today. Could you tell us a little bit about what motivated you to choose this topic?
Yeah, so basically, just having conducted my master's project, what I realized from, the people around me at the lab at Sainsbury Wellcome Centre and then the people I interacted with, it was really important to stress of a biological hypothesis or something that you're really specifically looking at in a biological sense in the brain. But what I realized from this is that this often leads many scientists to be very limited in what they actually search and research on. When I was conducting my research, which was basically on in situ hybridization, I had to create a new protocol or a new way to study the brain. And when I was doing this, I realized that my supervisors from the course, they actually thought that I wouldn't be able to do a good thesis because I wasn't looking at something biological or looking at something in the brain directly, but rather I was looking at a method instead. So what I realized from this is that we tend to think of successful theses or papers as something that can explain a biological region of the brain and what it can do in terms of a function. But I'm thinking that maybe we could, be a bit broader than this and look at neuronal circuitry in a more simple way rather than having to focus on a biological function for it.
Right. So do you mean that we should be focusing on looking for patterns or mechanisms that are more brain-wide rather than region-specific?
Exactly. So many professors, they spend a lot of the time in a specific topic or a specific region of the brain. So a lot of them become experts of the hippocampus or, amygdala.
Yeah.
And they basically say, we know everything we know about the amygdala or the hippocampus. But there could be a lot of similarities between the amygdala and hippocampus in sort of like neural circuitry, but they just aren't able to tell each other or communicate this because they aren't able to understand it in terms of the other brain region. If I were to take something like, let's just say from the hippocampus, like long-term potentiation, which we know plays a big role in memory, this is really just synaptic responses in response to neuronal activity. This can be done in any region of the brain, and it doesn't have to be tied down to memory. But when we conduct our research, we would like it to be presented as a theory of memory rather than just a theory of how neurons are able to interact with one another.
Yeah. I feel like that makes a lot of sense. And I would say that there are some circuits in the brain which can have parallels to other circuits, and the same principles can be applied to other ones. So, for example, the medial entorhinal cortex is divided into layers, and the top layer contains inhibitory interneurons, and also the remaining of the layers contain specialized cells that interact with each other. So the excitation and the inhibition result in a specific spatial cognitive function. And I think a similar parallel can be seen with the cortex, in that you also have the layer 1, which is also inhibitory, and then the neurons interact with each other. So comparing, for example, the cortex to the visual cortex to the medial entorhinal cortex, I agree that you could apply the same principles to both, or the same type of rules of connectivity to both. But for example, to pick at the amygdala, I don't know the specific areas of the amygdala, so I only studied the basolateral amygdala. But I don't know. I'm unsure whether this specific brain region, its circuitry can be applied to other areas of the brain, like the thalamus or the hippocampus, or like, for example, the dentate gyrus. Its structure and its connectivity leads it to have this very specific function of pattern separation that you studied, Clara. And I'm not sure whether these rules of connectivity can be applied to the whole of the brain.
Right, because just to expand on that, we did an previous episode just on the hippocampus, which is why I laughed just now when you said that. And I do love the hippocampus, but in that episode, we discussed how each subregion of the hippocampus carries out like a very specific function. So like you said, the dentate gyrus carrying out pattern separation predominantly, and then the next downstream region, CA3, carrying out pattern completion, and so on. So yeah, I agree. There are a lot of region-specific mechanisms. So how would you apply your thinking to this perspective?
Yeah, so basically, I think some keywords I want to pick out there is that you mentioned function and pattern recognition as like one of them. It does this specific thing. But in the end, like these are neurons, right? They're cells that are basically built to perform a certain function, and they don't really recognize whether they're performing pattern recognition or if they're performing something else. So I'm bringing this into the, the philosophical argument of like functionalism, where I'll explain this. So I hope that my explanation is correct, because many people would argue against it,
Specifically
obviously.
Philosophers.
Yes. But essentially, functionalism is that the way in which a thought, a feeling, or a certain behavior is made up, it is made by the functional connections that are created by whatever underlie your specific consciousness. So this doesn't have to be necessarily that your brain is housing consciousness. It could be any sort of medium or any sort of channel. Let's just say pieces of wood or rocks. As long as they create the functional...
Or
trees. Exactly. As long as they create the functional circuitry that is required for that thought, that feeling, or that behavior, they are able to realize it. So using this sort of like argument, what I thought a good parallel for this would be electrical circuitry. So electrical engineers, the way in which they build circuits, it's based off of very simple components. There's a power source, there is the wiring, the conductor, there is the resistance, and the way in which it works, essentially the objective is to power whatever is at the end of the circuit. So whether this is a light, whether this is a heater, whether this is a computer, it doesn't really matter to the circuit or to the power source what it is powering, as long as it just gets the power there, because that's what it's meant to do. So if you think about it like a one-bedroom apartment, right? You want to now provide a massive circuit which can allow all the power to be generated for a one-bedroom apartment. There's a certain way that you have to do it, right? There's a certain level of parallel lines that you have to create, serial lines that you have to create that will allow you to have the heater, the bulb, and everything to function correctly. So if an electrical engineer can kind of understand how this circuit is built, right? And what makes this circuit so good for this one bedroom apartment, and then say that, okay, how can we combine this one bedroom to be able to create a 3-bedroom apartment with the same circuit that can work just as efficiently? So this parallel is what I would like to take to the brain and neurons. So essentially, how can we say that the circuitry that is created is efficient and does the purpose that it needs to?
I really like that analogy you just laid out. I thought it was super clear.
Yeah.
And actually, to support it, I read this article from SWC where they basically interview Dr. Eleonora Russo, and she kind of talks to a similar idea of what you're talking about. So I'm just reiterate what she says. She asks the question, what coding structures does the brain use when engaged in different tasks? And are there hidden regularities in neuronal dynamics? And so she goes on to talk about that she believes there are recurrent mechanisms in the brain. For example, whenever there are oscillations or a certain connectivity structure between neurons that favor certain dynamics, these dynamics might lead to a specific function, whose content would then be determined by a specific brain region. And she's interested in finding a sort of coding grammar in the dynamics of neural activity, which is kind of what you were saying about this dynamics and the electrophysiology powering these specific brain functions and these neurons that are specialized to perform a specific function, and that then arises the different cognitive behaviors that we have, but powered by the same principles.
Yeah, this again speaks to an idea that we always return to in Neuroverse, which we first explored in our structure function episode, where Carolina and I were discussing like whether structure always equals function. And by discussing these concepts in terms of neuroscience, we realized that, well, in the brain, there's the structures and their functions, but the thing that kind of links it all together is the dynamics that occur between them, which is, again, what both of you are saying. Like the dynamics is kind of the underlying regularity, in the words of
Dr. Eleonora Russo.
Yeah, that can create the functions and links the structures to the functions.
Yeah.
Also, I wanted to mention that I loved the analogy you used in terms of like an electrical circuit, because neurons are actually modeled in terms of like basic electrical circuits, and you can computationally model a single neuron as an electrical circuit with conductors and resistors. And then you can create like models of a neuronal body and the dendrites as a more complex electrical circuit, and then you can create a model of two neurons together by then adding on top of this. And so technically you could actually model a simple neural circuit as an electrical circuit, which is often done in like theoretical neuroscience. So this leads me to my question for you. Do you believe that theoretical neuroscience is the way to go? Is it the most effective way of researching how the brain works?
Okay, I think theoretical neuroscience is the most effective way in being able to generate hypotheses and basically ask the question. But obviously, it is not the best way to find the answer. So we definitely need practical neuroscience. We need to understand brain regions and why they're so separate and all that for practical neuroscience.
But is the function of neuroscience, or is the purpose of research to answer the questions or ask the questions?
I think for me, it is to ask the questions.
Okay.
Yeah.
Because for me, it is much more interesting. And, you know, like what I would like to know is, what are we capable of rather than what are we currently doing? So that means, what is like, you know, the most complicated thing we can do? How can we drive ourselves to be, you know, even better, even faster, even smarter? And I feel like that is how we can achieve that or figure that out. And that is through theoretical neuroscience. You know, like we can make these circuits, and obviously, you know, in a physiological sense, there is probably a limit to where, you know, like how much a certain circuit can output at any given time. But in a theoretical way, we can actually find that limit and say, okay, what is the actual limit? How fast could we actually go? In what environment could these neurons perform even better? And I feel like that is what's really interesting about it.
Wow, yeah. I would definitely agree. I think somebody needs to answer the questions.
Yeah.
But in order to make research most efficient and valuable, you need to make sure you're asking the right questions.
Absolutely. I think in an age where we have like such incredible technologies like neuropixels, where we can record millions of neurons, it's not so much about whether we can get the data, it's about
how
and if how you analyze it, which questions are you asking, is this technique really relevant for the question you're trying to ask or answer? Yeah. So having talked a little bit about connectivity and functionality, and also given that we recognize the limitations of neuroscientific techniques, there was actually a really interesting study that is in bioRxiv, and that I think came out recently. It's by Sebastian Tremblay and Camille Testard. They're both first authors. And they conducted a study where they found that using a certain type of electrophysiological recording and focusing on the functional role of a specific brain area studied can lead to a cognitive misrepresentation. And so what they did is they looked at the monkey's prefrontal cortex, and they made it do 4 different types of cognitive tasks related to either visual discrimination, working memory, decision-making, etc. And for one specific task, I can't remember exactly which one it was, they stated that they could causally determine that through the neural recordings that this brain region, specifically 8A of the primate's prefrontal cortex, is needed for a visual stimuli selection task. They then did chronic neural implants where they recorded from the neural populations that they claimed were causally necessary for this visual discriminatory task. And they found out not only it wasn't causal, it was merely correlational, but that for the 4 other cognitively distinct tasks activated the same neuronal population. So they showed that it wasn't specific. And through their results, they showed that the methodology of neuronal recordings can be faulty, and that using only functionality to assess behavior, cognition, etc., can lead to misrepresentation. So in neuroscience, we can't just consider the connectivity. We need to consider also the molecular. We need to consider the behavioral. We need to consider all of these different levels and elements that occur in neuroscience. They proposed an explanation for their results, and which I found quite interesting, where these signals are simply feedback from other connected areas where critical processing happens.
Yeah.
And that ties into my favorite area of neuroscience, which is understanding like feedback and feed-forward connections, and how we're still grappling to understand and how to differentiate these different processes.
I think that ties into a really big problem in neuroscience, which is how we define cognitive functions. So obviously, as humans, we are limited by our subjective way of how we perceive how we think or how we are conscious. And so the breakdown of cognitive functions that we define, such as like memory, decision-making, etc., is all based on what we find useful or necessary as humans who are conscious. So our perception of the brain is very much tied to this because, like, historically, when people first started thinking about how the brain works, they used, is it phrenology?
Yeah.
Yeah. So basically dividing the skull actually into different regions and saying, oh, this part is necessary for language. This is for memory. This is for whatever behavior. And that's how our understanding of neuroscience developed across history, based on these cognitive functions. And so I feel like this is something we'll always be limited in through our subjective perception of how the brain works and thinks and functions. The starting point is ultimately, what are the functions that we need?
I think also, like, mentally, when we think about things, it's much easier to group them together and give them a certain definition of what it is. And then that just helps us understand it a lot easier. It's a lot easier to look at lobes of the brain, such as temporal, parietal, frontal, and that they have specific functions. When we know that that's not really the case, and there is a lot of interlink, and they, help each other in a lot of different ways, and they essentially share the load of, cognitive ability. They don't really have specific things that is just them.
But also molecular mechanisms or cellular mechanisms aren't generalizable to the whole brain. For example, in our episode on predictive coding and dendrites, we described like the theory of how dendrites integrate feed forward and feedback integration, and through the interaction of inputs within a single neuron, there's this form of predictive coding at a cellular level. But this only applies to specific neurons, excitatory neurons in the cortex that receive these inputs. And so then when we find a mechanism like this, we think, oh, well, since the cortex is involved in higher order functions, this mechanism is probably linked to this function because it would make sense.
It's very correlational, not causational.
But I mean, that is the easiest way to make progress, because if you just keep finding random mechanisms and not wanting to link it to the function, then you're not going to get anywhere.
Related to cognitive behavior and grouping it together, you made a really interesting point in preparation for this episode where you talked about animal models and how I guess it's related to the ethological relevance, and we assess how well or not they do a task based on cognitive functions we understand. Do you want to expand a bit more on that?
Yeah. Essentially, like, we look at animal models and how they complete a certain task, such as parenting or aggression. And, we have our own perception of what that looks like and what that doesn't look like. We can, even use computer models to be able to maybe get a better idea and, have it be a bit closer to the actual context of the mouse, but we will always have our own, perceptions of it. And we will group things, even though we don't fully understand them. We don't know why the mouse did that certain thing, but we will declare that, no, that is parenting because he licked the mouse.
And it's a behavior we recognize, right?
Exactly.
If a mouse does a discriminatory task and turns the wheel left versus right, oh yeah, he's making a decision.
Yeah.
And it's even like, this is why it's so hard to study learning, because you're like, oh yeah, the mouse's behavior in this task of finding food is improving over time. But how do you know if that's because it has learned the spatial fields of this area, or whether it has learned how to smell the food, or whether it why? We don't really know, but we link it.
And it's very like anthropomorphizing. We're projecting our human behavior onto the mice
in order to study ourselves. How ironic.
Yeah. So, and I think what I wanted to kind of like take this to now is that, okay, great. You've said how, the brain can be like an electrical circuit. We can take neuronal components and figure out that they do certain things in a certain circuit, and they may do a similar thing in another circuit, right? And this could be similar. But how does this help us, right?
Yeah.
I think the way in which this can help us is that when we think of like the stereotypical research paper, we'll have, how does the excitatory neuron in the lateral hypothalamus. I'm just being very general right now. Okay, in the lateral hypothalamus conduct this specific firing, like spiking rate for this function, right? Which is great. You've created your own niche. You've made your own research, which is completely unique to anyone else's, right? Which is good for you and good for research because we're branching out. But now, if another scientist is looking at the exact same circuit, but in a completely different brain region, they have no real way to find your research, and they have no real way to really understand what you've done because they are looking at something completely else. So if they search up something on the cerebellum, they're never going to find your research on the lateral hypothalamus. So I think that although it's great, that we create our own niches, we should create something a bit more generalized, a bit like a database. So we create a very simplified understanding, right, of our research. And we say that this is the circuit that it creates, this is the circuit that it functions in, and this is it. We can upload this into this database. Now, if someone finds a similar circuit, they can search it up to the database. Where do we see the matches? And then you'll find that, okay, this person worked in the lateral hypothalamus and he had a very similar neural circuit and this is what it did.
That's such a cool idea. It's kind of like the mouse brain atlas, but for connectivity.
Exactly.
Yeah.
Yeah, because I worked a lot with gene transcripts and like nucleotides, and I've found out that if you want to create a probe, you have to probe the gene atlas for the mouse. So when I did, you create a certain sequence and then you find the similarities of the sequences in all genes that you have. And you're able to see, okay, is it a 90% match? Is it a 50% match? And I'm thinking that if we can do this for genes, although it's a lot more simple for genes, right? Because we only have A, C, T, G, it's not that complicated. It may be a bit more complicated for neuronal circuits, obviously. But if we are able to create such a system, imagine how easy it would be for new scientists that want to probe a region of the brain that they don't know a lot about, where they can just take a circuit that they've already found through their own research and search it up and be like, okay, where has this been seen?
So what would be the constant factor that you would search up for the circuit?
Yeah, so
would it be like firing rates?
It would have to be a combination of a lot of things. It would have to be firing rate, it would have to be gene expression, it would have to be it would be quite complicated and quite comprehensive. But I think that, if we're thinking about neuroscience and how we're continuing to develop and we're really getting more and more complex, like year by year. In 10 years' time, we don't know how complicated our theories of neuroscience will be. Our theories just 10 years ago were a lot more simple than they are now. So I think that this could be something for the future that we could really think about, especially because our computers are also so much better now, and they will be so much better. So they will have the computing power to understand these slight differences in the neural circuit. So how can we create this as a database? I think that could be a really interesting idea.
I think that's really brilliant.
I find this idea very interesting. Something I did notice is that, so say you did have a database like this, in order to put any information or data on it, you would need to categorize it based on the location. So like the example you gave, you'd be like, okay, so I found this in the cerebellum. Let's put it under cerebellum. So isn't that further perpetuating the so-called problem of categorizing based on brain region?
Yeah, so I think for this database, what I specifically don't want to do is categorize it under a specific region. So what I would do is that you would have your paper. You can have your paper which fills that specific niche of the cerebellum, but you would then put a non-biased, just a circuit, and you would upload that onto this database. It will have a link back to the paper that you've written, but it would be just a very simplified diagram of neuronal circuits.
Okay.
But this neuronal circuit would inevitably be connected to anatomical areas. If I'm connecting the MEC to the amygdala to the hippocampus, it's still focusing on localized brain areas. It's a circuit and it has its own connectivity rules, but it's still related to anatomy.
Yeah.
So how would you separate it? How would you separate the connectivity from the anatomy?
So the way in which I think about this is that I'm sort of taking neural circuits and going like, okay, how do we study them kind of like, in situ? How do we study them, in other areas? We basically create like fake neuronal circuits that aren't actual, anatomically based or physiologically, capable as in a brain. But we're still able to study this. We're still able to look at how they interact in a very isolated manner.
That's like the principle of neural networks though, right?
Yeah.
Yeah.
It is pretty similar. And I think another point that I wanted to make was that, a lot of people would say, okay, how is this different to the connectome, right?
Yeah.
Mhm.
The connectome, this is where I think it's really different, is because the connectome project, from my understanding, really wants to understand how can we basically understand every single input output in the brain, and how can we get a sort of basis model of the entire brain.
Mhm.
But for me, that is not the goal of this. I think the goal of this is how can we understand it? So I would think that being more of like a macro scale, where it's like you're looking at the brain as a whole and every single circuit. I would take it down to more of a mesoscale, so not the micro. Mm-hmm. So micro would be that you're looking at specifically molecular markers, looking at, like very specific neurotransmitters, something very small. But in the mesoscale, what I would say is that how can we just look at as simplified circuits? So it's not human, it's not mouse, it's just a neuronal circuit. Because obviously, there are differences between mice and human, whatever, but I would say the very simple neuron, right, is pretty similar between the two. So if we can just have these understandings of neural circuits and how they work, we won't be even limited to something like a human. We can look around at different species and see where this is similar.
How is that different from just understanding and characterizing neurons and understanding their properties?
Or like coming up with mathematical theories for like dendritic function or plasticity rules.
Yeah, like we understand what an interneuron is. We understand the function and the connectivity of a pyramidal neuron. Like we understand neurons individually.
I would say that our understanding of neurons, the way in which we know them, are very isolated. So we understand an excitatory neuron by its own, like function of it being excitatory, right? Or an inhibitory neuron by being inhibitory. But once we start creating complicated circuits, which, combine excitatory, inhibitory, you have astroglia involved as well now, right? You have so many different cells that are playing and interacting in a specific role. We then get confused about what it actually results in. When we think about the models of circuits that we know in Alzheimer's, Parkinson's, we do understand that there are specific excitatory inputs, specific inhibitory inputs that are completely separate channels. We understand that, they play certain roles, but when it goes wrong, like in Parkinson's, we don't know which specific part yet, right? We don't know what is the exact area that shifts, that causes that difference. So what I think the difference between the two is that we can look at something in a very, isolated manner, like a neuron, or we can look at it in a very, holistic manner where we also take into account of the behavior. But I want to do something in the middle. Yeah. Where we just focus on the system, the circuit.
But for example, you can have a circuit that is excitation, very simple, or inhibition, or inhibition excitation, or excitation inhibition, or inhibition. Yeah. Would you just then basically create a database of the possibilities, in the same way that with theoretical neuroscience you would do, and you would just have a series of types of connectivity, as we mentioned, inhibition or excitation, or would it be more specific than that? Or how would it be different?
It would be that probably someone would upload a very simplified understanding of just excitatory, then inhibitory, or excitatory by itself or inhibitory by itself. You'll always have that. Because that's a very simple understanding of a circuit, that will be there. But I think it just helps that when we get to the more complicated circuits, that, where can we relate this? How can this be understood? Because some scientists, they focus on such a specific brain region, and they then think that when they found this certain circuit, that this has not been found anywhere else, right? This is so important just for the hippocampus and nowhere else. But if you really take the circuit out and then you start probing other regions of the brain, you'll find that there are similarities. You'll find that there are other parts of the brain that have long-term potentiation, and that it leads to a certain causal function. So where is this seen, and how can that help us in terms of our research? Because it would enlighten different regions of the brain and tell us that, okay, this is not that unique or specific to the hippocampus, but rather it can also be in other areas.
I like it.
Yeah.
I'm on board.
I think it's a really good idea.
Yeah.
I think that's the perfect place to wrap up the episode. Thank you, Subham, for joining us to chat about this, and really, really interesting pitch you got there.
Yeah, thank you, Subham. It's been super enlightening.
No problem. Thank you for having me.
Also, feel free to follow us on Twitter so you can get our latest updates, and support us on our Ko-Fi page, where you can donate to help us improve the quality of our recordings.
And check out our website for more resources on our blog. And also, we created a Google form sheet that you can give us feedback on how you would like to see the podcast improved and any other ideas that you might have. Thank you.